Welcome to today's session of the East Africa Journal of Neurological Sciences Club Bootcamp. So, my name is Lee Gitao. I am a level 6 medical student at the University of Nairobi, and I will be your moderator for today. I am also representing XYZEZ Med. So, the East Africa Journal of Neurological Sciences Journal Club is a collaborative initiative by the East Africa Journal of Neurological Sciences, together with JOCAP, JOGECA, and the Annals of African Surgery and the Anatomy Journal of Africa, in partnership with XYZEZ Med, with SCORI, with the University of Nairobi Research Club, CUMREC, and the Students Surgical Forum. So, through this bootcamp, our aim is to strengthen scientific critical appraisal skills, to promote engagement with local and regional journals, and to stimulate greater interest in research publication and peer review among medical students and early career researchers. For today's session, we will be focusing on the critical appraisal of an experimental animal study. So experimental animal studies play a crucial part in biomedical research, particularly in advancing our understanding of disease mechanisms, pathophysiology, and the preclinical evaluation of interventions before translation to human studies. So these studies allow for controlled experimentation while addressing ethical concerns that may not be feasible in human subjects. Our session will run for approximately 1.5 hours, and it will be structured as follows. We will start with a 15 to 20-minute presentation by one of our partnering organizations, summarizing the animal study and a review, including the study design, the animal models used, the interventions, the outcome measures, and the key findings. From there, we'll proceed to a 30-minute open discussion, during which we will collectively appraise the study, focusing on internal validity, appropriateness of the animal model, experimental controls, ethical considerations, strengths, limitations, and relevance to human disease and clinical practice. Finally, we will get our closing remarks and expert insights from our guest editor, Jimmy Gakure from the Anatomy Journal of Africa. So we encourage everyone to actively participate by sharing questions, critiques, and reflections. Feel free to engage openly and respectfully. This is a safe space for learning, designed to accommodate all levels of experience. So with that, first I'd like to invite Felix before we can invite our speaker for today.
We'll be using the EAJNS website now for all bootcamp related activities. So for the announcement of the papers, for the links to the articles and anything related to the bootcamp, so you can just find it on the website. That's to make it easier, and to also drive a bit of traffic to our website. And for the past articles, past sessions that we've had, you can find them on this tab, the EAJNS Journal Club tab on the website, and you can find all the relevant materials for the different sessions. So I know this caused a bit of confusion. I was informed that people did not see the link, so they thought there was no submission on the email. So this is the update. So just maybe just go to the website and familiarize yourself with it. And yes, so in case you have any concerns, you can always send an email, or just maybe you have my number, you can just text. So as Lee has mentioned, today's session is continuing on experimental studies. So we did randomized control trials. So for this session, we'll be doing experimental animal studies, and we hope to have another lively discussion today as well. So I hope Dr. Niva has been able to join. I can see she's not yet able to join. So meanwhile, we can have a bit of an open session on if you have had any concerns with sharing your work, or just how this particular bootcamp progress so far. You can just share any insights so far on anything we can improve on, or anything we can do better, and anything we're doing well. We welcome any remarks as we wait for our speaker to join. Um, she's joining in a minute. Thanks, Clarence, for the positive feedback. Yes, we try to send reminders as much as possible to keep you on track. It's very easy to get derailed. The attrition rate has been quite high so far. Started with really huge numbers. They've been declining over the over the past 2 months, which is expected. It's not easy, but we hope that you persist through the entire 6 months so that you actually gain the expertise that we intended this bootcamp to impart. And yes, keep joining the sessions, keep submitting your reports on time. But if you're not able to, we keep the forms open. So all the sessions forms are still open. So we'll close them at the end of the bootcamp in June. So after that, as we are awarding certificates, and only those who are able to submit for every single session will be awarded certificates of completion for this bootcamp. So kindly take note of that. And for those who've fallen behind, kindly maybe try and catch up with the past sessions. So I think, let me just confirm if she's joined now. Yes. So our speaker has joined, and Lee, you can introduce her and then we can proceed from there. Thank you.
Felix, thank you. So our speaker for today is Dr. Niva Mutungi from Skori. Doc, you can unmute and you can begin.
So thank you so much for this opportunity. I will take the least time possible because time is not on our side. I don't know if the topic has been introduced. So we've been having a bootcamp, a research bootcamp, and we've been having proposal appraisals, and we are critiquing proposals. So today's topic is on therapeutic potentials of Baronia amygdala, acetamide-induced neurotoxicity. Yeah. So at first glance, this topic looks complicated and very difficult. I don't know what level everyone here is. Are there students here? Are there doctors here? I don't know. Maybe you can share in the chat box so that we can know what we are dealing with. But simply, this topic is trying to discuss the therapeutic potentials of a certain medicinal plant, commonly called bitter leaf in Africa, in thiacetamide-induced neurotoxicity. So thiacetamide is a chemical. All right. So as I was saying, I was trying to simplify the topic because when you look at it, it can look complicated. Yeah. So therapeutic potentials, it's really a study. We're trying to look at therapeutic potential. Actually, not us. The person who wrote this was trying to look at the therapeutic potential of a medicinal plant with a difficult name, Vernonia amygdalina. In Africa, it's called the bitter leaf. Yeah. So he was trying to explore its therapeutic potential in thioacetamide-induced neurotoxicity. So I was saying thioacetamide is a chemical. Yeah, we wouldn't interact with it daily in our dietary, in our food drinks, but people who work in labs, people who work in industries, interact with so many chemicals, and thioacetamide is one of them. My name is Dr. Esther Niva Mutungi, MHB, graduate from Ozim University, class 2025, and I am so glad to have you today. So let's try and see what this topic is all about, right? So the abstract in short, we're seeing that the chemical is hippocampal neurotoxicity and microglial activation, modeling hepatic encephalopathy-associated brain damage. Now, we're tackling the abstract bit. I hope you hear me and see my slides. The abstract will summarize research. correctly. So we have said that this chemical induces hippocampal neurotoxicity. How does it do this? So this chemical usually causes hepatic. When we have hepatic injury, at times we have complications like hepatic encephalopathy. So an issue with the liver causing an issue in the brain, and hippocampus is part of the brain, and we can have neurotoxicity of certain regions of the hippocampus. Yeah. And all this neurotoxicity will activate microglial cells, which cause inflammation in the brain. Okay. So this study is trying to look at how the medicinal plants can be used to reduce the chemical induced neurotoxicity. So basically, how does this plant do this? Or how does the scientist who came up with this think that this medicinal plant reduces this neurotoxicity? So it's basically by uh, interfering with inflammation in the brain. Microglial cells cause the inflammation in the hippocampus, right? So causing the neurotoxicity. But this medicinal plant, the bitter leaf, stops or interferes with inflammation. Yeah. So interfering with the inflammation, thus reducing or stopping or uh, getting rid of the neurotoxicity. Okay. So introduction, I think I have already summarized this. So the hippocampus basically deals with memory, yeah, and cognitive functioning, basically spatial memory. So this medicinal plant has anti-inflammatory and neuroprotective properties. So anyone in this meeting who can tell me any antioxidant, any naturally antioxidant or anti-inflammatory substance you know of. It could be something we eat. It could be something that you've had people get injected or given at the hospital. Yeah, you can text, you can send it in the chat box. Tell me about any antioxidants, any anti-inflammatory uh, properties or substances that you know of. Tell us for chat box. Yeah, vitamin E, vitamin C. Perfect. Yeah, you're all correct. Yes, this is great. Ginger cloves. Perfect. Yeah. So this plant also has anti-inflammatory properties. Okay. And let's see. I don't know if this image isn't so clear, but it's called the bitter leaf. Perhaps you could Google it and have a look at it. I don't know what you call it in your language, yeah, but this is what it looks like. It's bitter. It doesn't taste good. But imagine something that tastes so terrible can heal your brain, can prevent neurotoxicity. Yeah, it's interesting. So the methods used in this study. So what I am trying to do is to summarize this study, this research. Somebody else will come and critique it and tell us the limitations they see in this study because it's a research. Yeah, we are trying to query, we are trying to find solutions to problems. It's definitely not 100% perfect. Yeah, so somebody else will come and critique the study. So we have talked about what the topic is about, basically what the paper is about. Now, which methods were used in this paper? Basically, I don't know if I said it, it's an experimental study. It was done on animals, so experimental animal study, but it's basically a controlled interventional study. Okay. And in this case, they used male Wistar rats divided into 5 groups. So we can say is, what do you think the kind of sampling, what do you think, which method of sampling was used in this place? Anyone in the chat box, you can tell us. You can tell us. How do you think they got to choose or decide on which rats to work with? Yeah, which kind of sampling did they use? Okay, okay. So basically, generally, the rats were purposively selected, okay? But the sampling method was by randomization. Purposively to mean they'll definitely choose maybe healthy rats, maybe fat rats, maybe there's a criteria, the weight, the probably specific weight of the rat they wanted, yeah. But basically, uh, purposively choosing them in a randomization way. I hope it makes sense. So, this experimental study done on animals, after sampling the animals, they put them into 5 groups, yeah, so that we had a control group where distilled water was used, another group where the chemical thioacetamide was used, only 100 milligrams per kg, then another group where the chemical and our savior, yeah, our medicinal plant was used, an extract about 100 milligrams per kg was used. Then in another group, we had the chemical injected or given into the rats, plus an additional, the medicinal plant now, but a higher dose, 200 milligrams per kg. Then another group, we had the chemical plus vitamin C. So vitamin C is also an antioxidant, anti-inflammatory property. Yes. All treatments were given orally for 14 days, right? Um, yes. So they were given orally. And now, what exactly were they monitoring so that you can come up with a conclusion that this medicinal plant is really a savior or um, it's just a waste of time. Yeah. So they assessed neurobehavioral tests. They looked for anxiety and spatial memory. Yeah. They looked for biochemical measures. They measured glucose 6 phosphate dehydrogenase activity and glutamate levels. They also did histological tests using specific stains like H&E and crystal violet. So this stains usually stain tissues and cells, right? So here they were specifically used to stain the CA1 region of the hippocampus. So they were trying to see the architecture and see how the chemical and everything given orally was affecting the cells. Yeah. Then they also assess the immunohistochemistry. Yes. So IBA1 marker, which is usually very specific for microglial activation. Okay, so what were the results? So basically, exposure to this particular chemical that we've said can be found in laboratories or industries increased anxiety and also impaired spatial memory in the rats. Yes. So the rats or the groups that were treated with the medicinal plant showed a partial reversal of these behavioral impairments. Okay. So the glutamate and glucose 6-phosphate dehydrogenase level did not really significantly differ statistically, suggesting behavioral effects weren't where these measures alone. So you see, we could have other mechanisms or other processes going on and giving us these results. And so we can't really know if it is our savior or our miracle drug that is causing all this alone, or, and we shall see that in the limitations and the critiquing of this study. Basically, glutamate and glucose 6 phosphate are very relevant in this case of neurotoxicity and liver injury. So we see that glutamate is an excitatory neurotransmitter, right? But it's also very key in memory and learning, right? So if we have an memory, learning, and uh, effect, like impairment in the hippocampal function, then glutamate will also be impaired. Yeah. And then this enzyme, glucose 6 phosphate dehydrogenase enzyme, is also relevant, particularly in the liver, because I know you have heard of glutathione. Yes, it's an antioxidant, and this enzyme G6PDH is relevant. It is important in the production of glutathione. So when we have these low levels, then we have low levels of glutathione, then we have the liver injury, and we have oxidative stress taking place. You understand? Then the entire hepatic injury, then the hepatic encephalopathy. So these, the enzyme and the neurotransmitter are key when you're trying to assess the neurotoxicity. Yeah, so histology was also done. Yes. So the group that's exposed to thioacetylamide exhibited pyramidal neuronal degeneration and disrupted architecture of cells in the CA1 region of the hippocampus. Okay, so the medicinal plant, especially the highest dose, the 200 milligrams per kg, preserved the structure of the cells of the CA1 region of the hippocampus, closer to the control group, the one that was just given distilled water, right? Which is like closer to normal, kabisa. Then they also looked at microglial activation, probably using the marker that we saw, IBA1, that is very specific to this. Then we see that TAA triggered activation of the microglial morphology. Yes. Then VA, which is the medicinal plant, reduced microglial activation, suggesting anti-neuroinflammatory action. Yes. So I don't know how many of us have heard of this bitter leaf. Personally, I had never heard of it, but I am sure I may have seen it, but I didn't know that it has a name, a scientific name, yes. But from the research I did, it has been used for several years for different conditions, for diabetes, for arthritis, for you see, it's just because it has anti-inflammatory, anti. So the fact that now we have someone looking into this medicinal plant as a potential of not just being anti-inflammatory, but being anti-inflammatory in the brain, then it's a beautiful thing. Yeah. So the discussion basically, our chemical that is being studied, thioacetylamide, induces neurotoxicity via neuroinflammation and microglial activation, not just excitotoxic glutamate imbalance. Then the medicinal plant or an extract of it benefits relate to its non-antioxidant and anti-inflammatory phytochemicals. So histological preservation aligns with behavioral improvement, supporting neuroprotective effect at the highest dose of this medicinal plant. Right. So this is a very, very interesting topic. Very, very interesting research. And we are proud of the person who came up with it. Yes. And I would like to invite the next person to critique the study and tell us more about it. Yeah, thank you so much for this opportunity.
Yes, hello. Thank you, Dr. Nyiva. The presentation was very clear and insightful, and thank you for breaking down the very big words into understandable concepts for us. So, I would now like to invite Felix to take us through the appraisal portion of the discussion. Just as a reminder, for the participants who want to participate via the chat box, please remember to change the setting on the chat box so that you send your messages to everyone. Uh, Automatically, it does not send to everyone. It sends to the hosts and panelists only. So change the settings so that you participate. For those who want to participate via unmuting, you will lift your hand and you will be unmuted. Thank you, and Felix, you can continue.
Thank you so much, Lee, and thank you, Dr. Niva, for breaking down that paper. It was also, as she mentioned at the beginning, it's a bit of a scary title. There's so many big ones, but after she's broken it down and digested it, and maybe you also read it for yourselves, it's not that complex, but it's a relevant study. I think even in the time we're living in, I think there's been talk from the Ministry of integrating traditional medicines and herbal medicines in in standard clinical practice, and it all begins from animals, since you can't start by experimenting on human beings. I mean, okay, people used to do that in the 1800s, but that's not ethical. So, experimental animal studies are preclinical studies, and we appreciate the Anatomy Journal of Africa for giving us one such study, and we can now dive into the meat of this and just discuss and digest it. So, okay, Clarence has already raised his hand. Um, uh, yeah, I think but yeah, so we will just get started. So the first part is the title. So again, we encourage you to be active, use the chat box, give your thoughts. No answer is wrong, since appraisal is just giving your opinion pretty much. So looking at this title, do you think it clearly identifies what the study is about? Is it a good representation of what the study is actually telling us? And does it contain all the relevant information? Does it tell us that it's an animal study in the first place? Does it highlight the intervention, the exposure, the outcome, and all that relevant information? And generally, is it a good title? Beyond anything, is this title, okay, I said it's scary. It was scary for me and for many other people, but again, we'd like to hear your thoughts. The floor is open. Clarence, you can talk.
Yeah, I can talk. Thank you. So for the title, it was able to identify the exposure, which is thiocetamide, the intervention, which is the plant Vernonia amygdalina. The outcome was well, it being able to, like, the therapeutic potential of the plant on neurotoxicity in the hippocampus, in the CA1 hippocampal subfield, but he didn't clearly state the study being an animal experimental study, and the species of animal was also not stated. Yeah.
Thank you, Clarence. Anyone else can use the chat box as well. Do you think it would have helped maybe because not everyone who's reading, okay, I feel like the number of people who know such Vernonia amygdalina are very few, but so many other people will identify this intervention if they put maybe into brackets bitter leaf. I looked it up also. It's a common delicacy in Nigeria. There's stew made from it, juice as well. Yeah, but again, anyone else with additional thoughts? We can take two more before we move on to the critique of the next section. You can also comment on the authorship. Is it okay? Does it, you know, reflect all the author roles and the relevant information and the affiliations?
Hello.
Yes, we can hear you. And as we mentioned the last time, you can start by introducing yourself and saying your affiliation, maybe institution or what you do.
Thank you, Joroge, and I'm Dr. Abdul. I'm doing currently pediatric surgery year 3. Thank you for these sessions. They are very helpful, especially some of us who are not initially exposed to research. I'm really appreciating the whole team. So my contribution is, in addition to what our colleagues said, in the topic, is that it doesn't state the type of research it is, especially if it is experimental, but we can really induce from the topic. Yeah, thank you.
Thank you so much, Dr. Ari. So I think that's sufficient for this part of this section. So we'll move on to the abstract. But if you have any additional comments, the chat box is there. You can always combine it when you lift your hand up for the next session, for the next section rather. So looking at this abstract on your screen, does it have all is it structured the way we've highlighted even in other sessions? Having a background that highlights what the objectives are. Does it have a methods and a results and a conclusion section and relevant keywords that are mesh terms? So yes, you can give our feedback at this point, and maybe Lee, you can read the charts if there are any. And if you would like to unmute, please raise your hand.
Hello, apologies. There was a problem with the chat box. You can now chat to everyone. You can now change the option. Thank you.
Anyone with thoughts?
Felix, on the chat, Valerie has a question. The question is as follows. For the titles, many of the studies we've seen so far don't have the full title. So like there's always something missing. Is it just creative freedom and it's the body that matters? Do you reject manuscripts based on the titles? And journal reviews papers before publishing, is it okay to just leave half a title?
Okay, so many questions. Actually, our editor-in-chief has joined us today, so we can pass the question to Mwachaka if you'd be in a position to answer. But if not, I'll just take it. But Dr. Mwachaka, if you're able to,
Yeah, Felix, just proceed.
Okay, fine. Yes, so we have our guidelines and our recommendations, and even as we've described in all the critical appraisal guides that we've been sharing, a good title should have particular characteristics, and because that's the first thing anyone who's going to read your work will see, and so it should at least highlight the population and the intervention, and at least something relevant to the outcome and the study design, or just the objective of the study. But nowadays there's a lot of creative freedom in terms of how you can frame the title, because for long it's been quite rigid and unattractive, and to get people to read, some of the titles are quite creative and clickbait. ish, not bait, but you know how media how they do it. They like blogs, and they'll have a very catchy title that will catch your attention and make you read the article. But generally, the best title is, you know, this framework that we give, you know, that will tell you the population, the intervention, the outcome, and the study design, because that's fully descriptive of what the study is. And even by just reading the title, you know what the study is about. So maybe you won't fully get rejected, but you may get corrections after peer review, because yes. So thank you for the question, Valerie. So maybe Lee, you can read the other comments, and if anyone has a comment on the abstract, you can leave your hand. Said his hand. Let me just allow Cyrus to talk.
Yes, Cyrus's hand is up.
Yes, Cyrus, you can go ahead.
Hi everyone, my name is Cyrus, MBCHB6. So I'd like to comment on the background and objectives. So I can see someone had commented that they don't follow the rigid structure, but one can follow. So for the rationale and the hypothesis, it's clearly stated that the hypothesis of the study is that the hippocampus is rarely examined in hepatic encephalopathy, and now this plant has shown promise in neurodegenerative diseases. So the objective is stated that the intent is to investigate the neuroprotective activity of the plant against the induced damage to the CA1 region. Yeah, I think on the method, I can say I saw there was a comment on the sex and the strain. It was identified as a male Wister rats. It's not identified with the species per se with the scientific name. And also we can see details about the group allocation, that were allocated into 5 groups, and then each group had 6 rats, but it doesn't state whether it was randomized or not within the abstract. Yeah, and then we can see there are details about the comparator and the intervention. That is, the 100 milligrams, the 200 milligrams are compared against a placebo and a standard antioxidants. Then also they're able to identify the key outcomes. That is, it lists the neurobehavioral tests, the biochemical analysis, that is the glucose 6 P dehydrogenase, there's glutamate measurements, and also the immunohistochemistry assessments and the histological assessments. But it doesn't state whether there was any blinding.
Okay, thank you so much, Cyrus, for that detailed feedback. So yes, and I think it would also help for this abstract. You will also just put separate, like you just put maybe it's the reviewer, maybe it's the formatting for the journal, but it's always easier to read through an abstract quickly if it has like the main sections, you know, the background in bold or separate, you know, just in bold. It makes it easier. And thank you so much for your feedback. Cyrus, and maybe something we forgot back to the title. It's all because as we mentioned, the training is also to help grow the next generation of peer reviewers. And you know, in peer review, the most important thing, so that it's constructive feedback, is to tell the author that you're reviewing, you know, how to improve their paper, not just what is bad, but how to do it better. So maybe for this title, you know, we'd want to restructure it into something like, you know, neuroprotective, something, effects of this Vannonia amygdalina, into bracket, what was it called? Bitter leaf against thiocetamide induced neurotoxicity in Wister rats, an experimental animal study or something of that sort. That will make the title, you know, much better, easy to read. This is quite a scary heading because there are so many big words. Even CA1 hippocampal subfield is such a huge word. But thank you so much. Maybe Lee, are there additional comments on the abstract? on the
Yes,
chat
the
box. And then
the comments are coming thick and fast. I will mention the comments that we have not already discussed. So, like, Peter Manson says that the abstract provides a clear background rationale, states the study aim and identifies the intervention and comparator groups and summarizes the findings. However, it lacks explicit reporting of key design safeguards such as randomization or blinding. So let me give two. We do two, then we continue. Wambui Irungu says the abstract does not mention how the rats were divided into 5 groups or whether blinding was done. There is no mention of sample size calculation. Confidence intervals have not been mentioned, and the abstract mentions using GraphPad Prism 5, but in the methodology, it says GraphPad Prism 6.1 was used in statistical analysis. I think we do those two, then we continue.
Okay, yes, I think, thank you. And whoever has also caught that, that is impressive. You're very keen, meaning you read every single line of the paper. So I think we'll dissect that more when you get to the methodology, the previous comments, because they're all on the methodology, because the abstract is just a snapshot of what the study is about. Usually has word count limits. You can't write everything, but also writing a good abstract on its own is actually also like an art. It's something that takes practice. It's not that easy. It's not just writing a copy pasting sections of the full manuscript that you have. It also has to be written, with a lot of thought behind it. So yeah, so I think that's sufficient, and maybe we'll share resources for how to write an abstract after the session as part of the further reading materials for this session. So we'll move on to the introduction. So this is the section where we expect to have a background of what the study is about. So a background of what the study
Excuse
question.
me, Feli.
So, yes.
There are some questions here that we have not tackled before we proceed. There's someone asking about the word count, like you've mentioned. They are saying that they had 354 words, but we are aware we're supposed to have 250. So if you can maybe shed some light, and then all keywords except for Vernonia amygdalina are mesh terms. Weirdly, both Vernonia and amygdalina are mesh terms individually. I'm not sure. There's another one that says in the conclusion, the author wrote thioacetamide treatment. I think this should have been thioacetamide exposure. This is in the conclusion of the abstract. And then the last comment, in the abstract, keywords have been mentioned, but the abstract fails to mention the limitations encountered. Maybe you can shed some light on that.
Okay. Yeah, so well, where to begin? I think I've actually forgotten the question. So, but in terms of word count, it depends on the journal. So before you send in your paper for review, just always go through the author guidelines that are on every journal's website. Check the recommendation. But okay, 350 isn't too bad, but others prefer just 250 to 300, depending on whichever journal you're submitting to. So always just check that. And again, for the rest, maybe it's a challenge to put everything. If even limitations, typically they're not really included in the abstract, because the abstract is just a small snapshot and summary of what the paper is about, because that's what people, whenever you're gonna read a paper, you just read the abstract first to know that you'd want to read it more. So you can't include everything. So I think that's the response I'd give to those other two questions. But we'll see more in terms of the questions on the methodology and the conclusions and whether they are good. After we go through the entire paper, maybe towards the end we'll discuss that. So anyone with feedback on the introduction? Is it a good background? Have they given their hypothesis like a good enough meat to make it at least biologically plausible, like you understand how this can cause this and then this can reverse this, because that's exactly what they are doing. And have they highlighted the relevance of this study, why they're doing it in the first place and why it matters, and how this animal model can translate to human disease. So the floor is open. So just give your feedback. You can use a chat box or you can unmute, but it would be nice to at least have one or two people speak on the mic. Yes, Lynn, you can go ahead.
Hi, good evening. My name is Lynn Maina. I'm an MBCHB student at Kenyatta University, level 3. So I would like to give my comments on the introduction section. I found it fairly well detailed relative to some other areas. It met most of the expectations, like they give the rationale behind using TA, the fact that it gives enough time for studies of hepatic encephalopathy induced neurotoxicity in humans, and also there's the mechanism by which the thiacetamide induces the hepatic encephalopathy, and they compare that to how the hepatic encephalopathy occurs in human beings who are in acute liver failure. And the authors also described the effectiveness of their intervention, which was Venonia, and they back up a lot of the things that they discuss using studies, and they cite the studies. They also give a hypothesis, and their hypothesis is pretty much plausible and it can be tested. I thought that it would have been good to expound on how their findings could be translated into humans, like just actually saying it, not having someone infer. And also, they could have given more details on the species or the oyster rats, and perhaps why it was the most appropriate choice. Yeah, I also thought that they could have explicitly stated why, not the why, but they could have explicitly stated the model, the intervention, the comparators and outcomes. Like, you didn't really have somewhere to refer, you just had to infer. Yeah, those are my comments.
Thank you so much, Lynn, for the detailed feedback. That's very constructive as well. And yeah, especially because she's also included like what she'd want the authors to do, which is also very recommended. So Lee, you can read the comments.
Yes, there's one comment. On the hypothesis given in the introduction, Wangari says that she feels that it would be the best time to specify that the study was done in the CA1 subfield of the hippocampus, since they are not particularly studying the entire hippocampus.
Yes. Thank you for that. Any other feedback? Or can we move on to the methodology? Yes, there's a hand up. Let me just unmute it. Oh, I think it was Peter who raised his hand, but it has vanished. Or maybe it was accidental. Peter, if you meant to raise your hand, you can raise it again. I think it has disappeared from my screen. But I think, yeah, they try to include quite a lot of detail to make the hypothesis to try and formulate the hypothesis. First, it was a bit confusing even for me when I was trying to understand what it was about, but it makes sense somewhat. But in terms of how they framed it, they could have done it a bit better, just to make it easier to understand even for layman, because not everyone is is an expert in pharmacognosy and these things of extracting things things from plants for medicinal purposes. But I think that's good enough for the discussion on the introduction. So we'll move on to the materials and to the methodology. So this is the main part of the sections of the paper, because this is what the authors did. So you can comment on whether they described the study design well, and whether the study is appropriate for this kind of study question that they have. Did they have ethical considerations and animal welfare? That's very important, because you can't just torture animals without justification. And did they describe their animal model, in detail? Oh, this was a, male rat. This was the number. This was how much they weighed. They were all the same, or they were different in this way, that kind of thing. And how they were housed, how they were fed, how they were taken care of. That's important. How they were protected from stress, all that. And how they they had about 45 or 6 groups, different 5 groups. How all those groups, how was allocation done to those groups? Was it random? Was it not? Why was it not if it wasn't random? And was there any blinding? How did they give the intervention? How did they ensure it's standard? How did they compare? How did they standardize everything from comparison to just all those things? And how did they eventually assess the outcome? How did they measure? And what tool did they use to measure all these set outcomes that they were assessing? And the tools they used, are they valid? And did they make them up themselves? And if they did they describe how they came up with them and justified why? And did they have a sample size calculation to justify the number of rats? And the number of rats that they used, sufficient from a statistical point of view to give like viable and results for the study, and how they analyzed their statistics, how they came up with the results they're reporting. That's also important. Yes, among all those things, that's what the methodology should contain. It's quite wide, but you can just comment and share your thoughts based on what you read. Was this methodology good enough? And even the figures, are they good? Are the tables just anything? You can just unmute at this point and or use the chat box. Because they assessed a lot of things. They did biochemistry, they assessed the neuropsychiatry or neurobehavior, they did histology, they did neurotransmitters, they did immunohistochemistry, they did a lot. So, anyone with thoughts? Or anything in the chat box? Lee?
Yes, there's only one comment, and the comment is in reference to the introduction. Peter is asking why it would be good for the authors to mention why the CA1 subfield was the specific area of study.
Oh, that's in the background, yeah. I think they said, I don't know, they said something about it being involved in memory. Also didn't get very well, but they mentioned something. But yeah, if you read and it didn't make sense, it would probably not make sense to other people as well. So yes, so yeah, that's important, so that it's clear why the CA1 field of every other part of the brain, why that point. And this thing is going, TAA is going everywhere, not just there. But I think that's sufficient. Any comment on the methodology? At least you can get maybe 2. The methodology is always the main part we'd want to discuss. So Ita, let me just allow you to speak.
Okay, I have a comment about the intervention methods.
Okay, you can introduce
Because,
yourself.
okay, I'm Darwin, medical student at UON. Yeah, I have a comment about the intervention methods. Because when you look at Group E, there is an intervention that was not mentioned in the abstract. That is the 200 milligrams per kilogram of aluminium chloride. So it's the first time it's appearing, and it was not mentioned in the abstract. And its relevance to Group E, and also its relevance to the whole study wasn't mentioned. And if you also check in Table 1, it is also not included. Yeah.
Thank you. It's a good catch, Darwin, because it's come out of nowhere. It hasn't been mentioned at any point. So, and they ought to justify, and because that's a confounder, why have you given it? Yeah, compared to the other groups. So thank you for that. Lee, in the chat
Yes,
box.
Ellie, there is a comment on the chat. Eddie says that I feel the authors should have expounded more on sampling and allocation of the rat models in their respective groups, block or computer randomization.
Yeah, because they didn't actually say how they did that randomly, or even if they did it. So thank you for that. Any other feedback? Lynn, you can go ahead. You can speak.
Mine is going to be a bit more detailed, I guess.
Yes.
So I found this, generally speaking, the methodology section wasn't as rigorous as I would have expected it to be, because there are certain areas that are missing. So, for example, in the study design, under the study design, they did not mention explicitly which study design they used. Like, yes, they used the parallel group and the parent, but they should have stated that. And also, they didn't define the primary outcome and also the time points for assessing the outcomes. And according to the guidelines, they were supposed to define the unit of analysis and unit of, I've forgotten the other one. So without defining those, you can't tell whether there was any pseudo replication in the study. And then even as you go further, you will not see any explicit definition of that. So you can't really tell how they were assessing. Yeah. And then there was this section and replacement, reduction, and refinement and animal welfare. There's simply no mention of that, how that was handled, whether there was any anesthesia, yeah, all that stuff. For the animal model details, they stated that all the 30 Western rats were male, and they didn't really defend why they used male rats only. It could have been important to give the why they used that and not mix just female and male. And also they didn't define the exclusion and inclusion criteria. And they failed to mention whether all the 30 rats actually completed the study, and if there wasn't any that completed the study, how that was handled. The randomization and blinding part has been handled. I think they should have handled that better. For the intervention study, intervention and exposure part, the standard drug they used was vitamin C, but according to some digging, there are other more standard drugs that are used when someone has hepatic encephalopathy. So I felt that they should have also expanded on why they used vitamin C, they chose vitamin C over the other standard drugs. For outcome measurements, they were supposed to mention who was performing who was assessing, for example, who was conducting the biochemical assays, their expertise and training perhaps, because for instance, if a student who is not very experienced performs the studies, the accuracy of the results could be compromised. So I felt like they should have also commented on that. Sample size wasn't, they didn't show how they calculated the sample size. They just stated that the sample for each group was 6. And yeah. And then for the statistical analysis, as one of the commenters had said that there is a discordance in the software that was used. And also the authors do not really say how missing data was handled, if indeed it was there. Again, effect sizes are not reported, so you can't really appreciate the magnitude of their findings. Yes, that is all.
Well, thank you, Lynn. Maybe you can appreciate her by showing some emojis. That's very detailed. I think you've been the top performer for today's appraisal. You've carried it on your back, giving very detailed feedback. That's very good, and keep up the diligence. And I think, even as we continue to go through different studies, we get to see that it takes hard work. There's a lot of work that is required, and thoroughness to do a good quality study, and because that's the main objective, that we are able to really improve the quality of our research and output from this part of the world. And it begins by knowing what you actually need to do in the first place. So that's very good. So maybe the only addition that I'd give here is on the animal ethics. As you've had, you can't just buy your own rats and decide to do a study. There are guidelines for animal ethics, and there are ethical review boards. For the same way, you'd need to apply for ethical review for human studies. Like, okay, I am in UN, so I'll just say KNHUN, but there is one in Moi. There's Aga Khan, all those different ethical review boards. There is one for animals. There's one domiciled at the veterinary department of the University of Nairobi, and I think all these other universities or mostly learning institutions are the ones that have, but there are others. So, and Lynn also hinted at it, the R's of animal ethics. There were originally 3, actually now 5. There are for that usually good. There's a framework called the Arrive 2.0. You mentioned it in that template we shared. That is the international standard for how to do animal studies. And if you intend to do an animal study at any point, it's good to familiarize yourself with that, so that the work that you do is of the highest standard. So thank you for that, Lynn. So
Yes, hello, Felix. There are
yes,
some comments in the chat.
yes.
Wangari says that there's no mention of blinding in the methodology. I think we've said that. And then Joy says, is there a reason why TAA for Group B was administered for 14 days while all other interventions were administered for 24 days?
Wait, is that the reverse? Okay, because the control they gave it for. Wait, TAA. Okay, looking at the table, 14. Yeah, there's a discrepancy between the control and but there's something they said 14 days plus 10 days, respectively. Oh yeah, there are different protocols. can see there was one they were giving 100. Okay, Darwin has his hand. Maybe he wants to save us. Darwin, you're unmuted. You can speak.
It was unintentional, so
Oh, okay. Yes. So they ought to expound on why. Jemima, you can go ahead.
Thank you. On that, I think I answered in the chat, but I'll answer again. Group A was a distilled water, it was the positive control. Group B is the negative control, so it only received TAA only for the 14 days. Group C, D, and E also received TAA for 14 days, but they also received, Group C and D received VA for an additional 10 days, while Group E received vitamin C for an additional 10 days. Hence why their treatment days are coming to 24 days. But Group B is the one to show us the negative effects, the neurotoxic effects of TAA, so they don't receive the additional 10 days of VA. That's why it only has 14 days.
Thank you so much, Jemima. Yes, that makes sense. Jemima, you forgot to introduce yourself.
Hi, Jemima, cardiothoracic resident here for University of Nairobi.
Thank you for joining us. So, Lee, I think this is sufficient for the method. Just to tell me the additional comments on the methods before we move on to
the rest of the comments are just telling us what Dr. Jemima said. They are trying to answer the question posed by Joy, so we can proceed.
Okay, so for the results, let's continue, pick up from there. So do they have all the main parts of the results? And have they reported what happened to the animals? Have they given started with how comparable the animals, the groups were at the beginning before any intervention? Have they reported their primary and secondary outcomes well? Do they have the good statistics and p-values, confidence intervals, all that stuff that comes with reporting results? So anyone? And what do you think of their tables and graphs and images and that is included in the results? You can use a chat box or unmute. Yeah, we can take maybe 2 because we're trying to not get to 9 p.m. Anyone who'd like to unmute and also use the chat box? I'm scrolling through. But all in all, we appreciate the author for all the work. You can imagine just going through the paper, how much work went into this study. It's a lot. So
good evening.
yes, it's good. Yes, good evening.
My name is Paul Muthuya. I am a medical student. Yeah. For the results, the paper implies that all 30 rats, which was the beginning number we had, they all made it to the end and their number remained consistent, which might raise some questions given the toxic nature of thioacetamide. So that the results do not mention whether there were deaths, which is expected given such a procedure. So, and they do not mention any exclusion criteria if any animals died.
Thank you, Paul. Thank you for that. Anyone else with further thoughts on maybe some other parts of the results? They can also be positive.
There is a chart from Clarence. He says that the primary and secondary outcomes were not clearly defined.
Yeah, I agree. Okay, yeah, but they were assessing multiple things. And okay, at least they clustered them, the neurobehavioral, the histology as well, among other parameters. But it's always good to state them explicitly so that we know what the main takeaway is. Thanks for that. Any
Clarence
additional thought?
Clarence continues and says there was no animal flow diagram.
Yes.
And then Wangari says the study did not also mention any adverse events or protocol deviations.
That's very important. And if we didn't have any, it's good to mention to make for people to ask themselves if there were. It's good to just say it before, yeah. So that's good. Anyone else, or we can move? Can even say the tables look nice and colorful. That's also feedback. These graphs rather. Okay, I think that's okay. If you have additional thoughts on the results, you can always come back and just highlight them. But I think they were pretty decent, and also the photomicrographs of the histology of the hippocampus looks nice. And yeah, they're well labeled. They have arrows and all, and yeah, so that's also good feedback to give. And they have descriptive legends, and they have a scale, which are all important parts of when you're reporting photomicrographs. They should be good quality and also be well labeled, have descriptive legends, and also have a scale. That's all. And so the scale is here, and so what you can use it to measure. So that's good enough. So for the discussion, we can also fly through this section. Anyone with feedback on the discussion? So yeah, was the discussion, did they interpret their findings in a logical way that's also biologically plausible? Did they address any areas of bias that may have come across? Have they described how this, whatever they did, mimics human disease and how it translates to clinical applications? And also just future recommendations and directions. And also we'll combine the conclusion and references as well into this part. Are the conclusions good? Are they, so are they plausible, and are they supported by what the study did and what they found? And does it have like a takeaway message for the readers? And are the references good as per the journal's guidelines? And are they appropriate? All that stuff. So this is maybe the last feedback part of the session, and then we'll go to our guest editor. So anyone with feedback, use the chat box, unmute.
Yes, we have Wangare who says the study managed to demonstrate the neuroprotective ability of Veronia amygdalina. The study also managed to demonstrate how it mimicked hepatic encephalopathy and how it is induced by TAA, but did not mention differences in species, strain, age, and selection.
Thank you for that, Wangare. Any additional, anyone else? Feedback on the discussion? Take one more.
Another comment from Lorna. She says that in the discussion, they transparently acknowledge that the active constituents of the plant extract were not isolated, which limits the ability to define the exact mechanism.
Thank you. Thank you for that. Maybe you can take one last one, and then we can wrap up the appraisal part of this session.
Yeah, I can do the conclusion. So yeah, the conclusion, it's able to clearly state like what was demonstrated in the models, and it also avoids making unjustified clinical recommendations, and also is able to identify the next steps. So in general, the conclusions are supported by the data in the study. And then for the references, in the guidelines, I was saying there should be a citation for Arrive 2.0 guidelines. There was no citation for that. Most of the references were current, but around 11 were not. They were more than 10 years old. And then the 31st citation, if you were to scroll down, it seemingly doesn't exist, but I just found people referencing it all over the web. I could be wrong, the 31st one. But other than that, all the other references exist. Yeah.
Thank you. Maybe it got redacted. Maybe the paper, sometimes journals, or maybe the journal wasn't working because maybe it stopped. Yeah, but thank you so much for also going through the references to confirm that all of them exist. That's very important, especially in the current day and age, where there's a lot of AI use, there's a lot of fake references that you can find authors putting. So it's always very important to confirm that. And you on that same note, from the most recent publication issue from EAJNS, you can read the editorial. It contains more information on our updated policies and guidelines on the use of AI for writing manuscripts and everything to do with that. So I think at this point, I just want to appreciate all of you for, again, a productive and wonderful session right from the beginning till this point. So we've had another wonderful discussion. I hope you've learned. And yes, you can always come back and review this when you want to do your own study in the future on animals. So at this point, I will hand the session back to Lee, who will now invite our guest editor, who will give his insight on how the session has been, and maybe just a few pointers for us as we continue doing our research now and hopefully in the future. So thank you all. Back to you, Lee.
Yes, thank you everybody for contributing to our discussion. Thank you to Felix again for leading our discussion, and to everyone who has bravely unmuted their mic or written their comments on the chat. So I hope we've all learned from each other, and we've all learned how to appraise animal studies further. So now I want to invite our guest editor from the Anatomy Journal of Africa, Jimmy Gakure.
Good evening, everyone. My name is Jimmy Njoroge. I'm a level 6 MBCHB student at the University of Nairobi. Today I have the unique privilege of wearing two hats. I have the privilege of working with Esther in SCORI. I'm a part of SCORI. And I am also the immediate former managing editor of the Anatomy Journal of Africa. So thank you everyone for collaborating on this wonderful project and discussing one of our publications. This was a paper that we published in 2024, in April 2024. And you have correctly and rightly highlighted some of the strengths of the paper and some of the weaknesses as well. Maybe I could give a few insights with regards to the studies, study design, as well as the challenges associated with it. This study is a perfect example of a quasi-experimental study. It's quasi-experimental because the principal investigators did not have full control of the study variables. We do not really know where they got the rats from, if they all had the same genetic makeup. We know oyster rats are usually inbred, but sometimes if they come from different places, that could imply different genetic makeups. But it's an assumption in most quasi-experimental studies, and it should have rightly been mentioned within the methodology. But a key weakness of most animal studies, particularly quasi-experimental ones, apart from what I just mentioned, is the number of factors or the number of mistakes that can occur during the study. You have rightly mentioned a million things that were not covered in the study's methodology, in the study's results. We could keep discussing other weaknesses, like if we assume that thiocetamide induces neurotoxicity through the hepatic encephalopathy route, then you'd have to ensure that hepatic encephalopathy has actually occurred in these rats, meaning that we'd have to check for those markers. I don't think that was covered in the methodology. So these are examples of challenges that we have with animal studies. And if you check our issues now, you can check our website, anatomyafrica.com. That's AJA's official website. You'll see that we don't really publish as many animal studies now. That's 2025. I think our 2 issues last year did not really have as many animal studies as the previous years. And it's because of some of these major weaknesses and major oversights that most authors tend to have while doing their studies. Even this study, if it had been submitted this year or last year, we would have probably rejected it. But this was 2 years ago, so they were fortunate enough to fall through the cracks. So thank you everyone for being so vigilant and being so keen. I welcome you to join our editorial team also. We need a lot of peer reviewers to properly review our scientific papers. We don't just publish animal studies, we publish clinically relevant anatomy. So if you have anatomical variations you come across or clinically relevant anatomy in animal studies, as long as they're very robust, we will publish them. So feel free to, if you have any research that's anatomy related, that's human anatomy related, not animal, feel free to submit it to our journal through our website. I will type it also on the chat box. So I've just given a few insights on that. Thank you very much to EAJNS for offering this wonderful platform in this bootcamp. Thank you to Scory for the wonderful presentation. Thank you to, I guess I'll pat myself on the back and say thank you to Aja for the paper that was discussed today. And thank you to everyone who participated in the discussion. And even those who didn't, thank you for listening in.
Yeah, I believe we've come to the conclusion, unless Felix, there's something else.
No, I think that's it. Just appreciation for all of you and to Daktari Online who've been our hosts and have been providing the platform. So we'll share the next paper. We shall be looking at a quasi-experimental study for session 7. So just be on the lookout for your emails, which will direct you to our website. So thank you all. Have a good night.